Infection with hepatitis C virus induces a wide range of adaptive and innate immune responses. In some individuals, these immune responses are successful, which leads to permanent control of HCV infection, referred to as spontaneous HCV clearance. However, in most instances, immune responses are not successful and chronic infection persists. HCV therapy permanently eradicates infection in some individuals. However, in many cases, HCV therapy fails to eradicate infection and the chronic hepatitis C persists. These very distinct outcomes, despite similar exposure to the pathogen, strongly point to host genetic determinants for the control of HCV infection. Logically, previous studies investigated candidate genes that were known to influence the pathogenesis of HCV infection. However, most genetic associations were not replicated. Fortunately, it is now possible to systematically screen the entire human genome for common genetic variation associated with the disease outcomes. We performed a genome-wide association study in 1,362 individuals with chronic and spontaneously resolved HCV infection. We genotyped over 500,000 SNPs per individual. Study outcomes were spontaneous HCV clearance and response to HCV therapy. The Manhattan plot shows the p-values for the association of single nucleotide polymorphisms with spontaneous HCV clearance in the top panel and with response to HCV therapy in the bottom panel. All associations that reached genome-wide significance mapped to the IL28B gene indicated by the red arrow. This finding was consistent for both outcomes, spontaneous clearance and response to therapy. Resequencing of the IL28B gene identified distinct haplotypes that were associated with the clinical phenotype. IL28B encodes interferon lambda 3, a type 3 interferon with antiviral activity. Our findings are in line with very recent studies that reported a strong association of genetic variation in the IL28B gene with response to HCV therapy and with spontaneous HCV clearance. Our findings underscore the role of interferon lambda and of innate immunity for the natural and the treatment-induced control of HCV infection. Our results could have a substantial impact for prognosis and therapy of HCV infection.